Thank you for the invitation to speak at the workshop. And thank you all for joining me today and taking the time to learn about building brain-wide models. So over the past decade, neuroscience research has made remarkable progress, but our understanding of the brain is still fragmented. Recent efforts by the International Brain Lab have taken a major step forward by building a brain-wide map of neuroactivity across many animals. However, building a unified model of the whole brain is challenging because we can't simultaneously record from every region at once. To overcome this, we need to seamlessly stitch together data from different animals and brain regions, filling in the missing pieces to have a more complete picture of the brain. So to do this, we need a foundation model that can integrate and generalize across diverse brain regions and animals, allowing us to derive general principles of neural computation. And today, I will explore how to build towards such a brain-wide foundation model. So previous efforts like Drag for registration, DartSort for spike sorting, and Lightning Pose for pose tracking have improved IBL data quality over the years. Building on this, our goal is to build a universal translator for IBL data, one that can seamlessly map neuroactivity across spatial temporal scales, link neuroactivity to behavior, and support downstream tasks such as brain region decoding and cell type classification. To do this, we take inspiration from self-supervised learning, where models are trained on large amounts of unlabeled data during pre-training before being fine-tuned for specific tasks. So this talk is structured as follows. In the first part, I will talk about how to build a foundation model capable of learning generalizable representations across different spatial temporal scales of the brain. In the second part, I will present our recent work on a multimodal multitask model designed for simultaneous neural encoding and decoding at scale. So recently, we have witnessed the emergence of many neural foundation models. However, most existing large-scale models are either supervised or overlook the intricate relationships between brain regions. For example, Ryoo is a multi-session model that predicts behavior from neuroactivity, but it does not model activity dynamics of brain regions. NDT2 uses self-supervised learning for activity prediction, which is similar to our approach, but it does not consider region-to-region interactions. While MD-like focuses on multi-area interactions, which is similar to our objective, it does not scale across multiple animals and brain regions. And we argue that a neural foundation model must be able to translate across all spatial scales of the brain, including population level, region level, and single neuron dynamics. So to do this, we propose multitask masking as a pre-training method to teach the model about the spatial temporal structure of the data using a mass modeling approach. Traditionally, mass modeling is performed by masking and then reconstructing neuroactivity in random time steps. While this teaches the model about the temporal dynamics, it can overlook important spatial structures in the neural data. So to address this, we propose 4 masking schemes to capture the diverse patterns in the data. The first one is called co-smoothing. For this one, we randomly mask neurons and reconstruct their activity using the unmasked neurons as context. This allows the model to learn how individual neurons relate to the full population activity. The second one is causal prediction. Similar to a GPT-like model, we mask future time steps and then predict them using past time steps. The third one is interregion masking. For this one, we randomly mask neurons in a randomly chosen brain region and then reconstruct them using unmasked neuron in other regions as context. This allows the model to learn cross-region interactions. The last one is intra-region masking. And for this one, we also randomly mask neurons in a randomly chosen brain region, but then reconstruct them using only unmasked neuron from the same brain region as context. And this allows the model to learn intra-area dynamics. So we think that a model trained with all these different masking objectives will be able to solve many different downstream tasks and inference time. And to train MTM, we randomly sample a masking scheme for each batch of the input neural data, and then mask the data according to the sampled scheme. And to make sure the model has seen different examples of each masking scheme, we alternate across these masking schemes during model training. Then we pass the masked neural data to a tokenizer to create the neural data tokens and use a learnable prompt token to tell the model which masking scheme is being applied. This prompt token can be passed to the model and the inference time to adapt the model to the corresponding downstream task. We then feed the tokens into a transformer model to infer the firing rates, which we can use to reconstruct the neural activity by assuming a Poisson observation model. So for the experiments, we use the IBL repeated site dataset, which includes neuropixel recordings targeting the same 5 brain regions in mice performing the same viral decision-making task. And we use trial-aligned spike-sorted data from 39 mice. And we bin the neural activity using 20 millisecond time windows and fix the trial length to 2 seconds. To evaluate how well a neural population model generalized to different downstream tasks, we designed a number of unsupervised and supervised tasks. The unsupervised tasks include co-smoothing, forward prediction, interregion, and intra-region co-smoothing, which roughly correspond to the proposed masking schemes. And we use bits per spike as a metric to evaluate the prediction quality for each neuron. The supervised tasks include choice decoding and whisker motion energy decoding. And choice is a binary variable, so we use accuracy as a metric, while motion energy is continuous value, so we use R² as its metric. And as I mentioned, mass modeling is usually performed by masking random time steps of the neural data. And what we call this masking scheme temporal masking, which is our baseline. To understand the importance of each proposed masking scheme, we train the transformer in a single session using each individual masking scheme and compare them to model trained using the baseline temporal masking. And we show the ablation results in this table. So we can see that each masking scheme improves its corresponding downstream task over the temporal masking baseline. And this shows that each masking scheme can teach the model about a different aspect of the neural population. In the scatter plots, we compare MTM to the temporal masking baseline across all 39 animals. And we see that MTM outperformed the baseline in all neural activity reconstruction tests. And this tells us that how training with diverse masking schemes can lead to a more structured understanding of neural activity. Since our goal is to train a large-scale model across many animals, we need to allow multi-session training. And one challenge is that different animals have different number of neurons, so the neural data from different animals have different feature dimensions. To address this, we use session-specific stitchers to map the input data with different number of neurons to latent representation with the same dimension, which is then passed into the transformer. We also use session-level context embeddings to distinguish the data from different sessions. So although the identities of the neurons are changing across sessions, we think that by training across multiple IBL repeated site datasets, which share very similar anatomical structure, MTM should be able to generalize well to unseen animals. So we pre-trained the transformer using MTM and the baseline temporal masking on data from 34 sessions. We then evaluate their performance on 5 held-out sessions by fine-tuning these pre-trained models on each held-out session. So in the scatter plots, we compare the fine-tuned MTM and the fine-tuned temporal masking baseline. And we see that MTM outperformed the baseline across all activity reconstruction tests, which shows its generalizability to unseen animals. So far, I have only shown the numbers, and in case you might be wondering what the beast per spike metric means, we compare the prediction from the fine-tuned MTM to those from the fine-tuned temporal masking baseline. And in the figure, we highlighted areas where you can qualitatively see that MTM outperformed the baseline. For example, in the inter-region test, the baseline overpredicts the firing rate, while in the intra-region test, it underpredicts. We also perform a scaling analysis across 110 and 34 pre-training sessions and compare the fine-tuning performance of MTM to the temporal masking baseline. So for MTM, increasing the number of pre-training sessions improve performance on all downstream tests, except for choice decoding, which saturates on 10 sessions. And for temporal masking, performance across all downstream tests saturate after 10 sessions, except for forward prediction. So this shows that MTM is a more promising approach than temporal masking for scaling neural population models to multi-animal and multi-regional data sets. So that concludes the first part of the talk, which focused on understanding the relationship between brain regions. Now, moving on to the second part, I will present our recent efforts to characterize the relationship between neural activity and behavior. So the IBL brain-wide map project tries to understand how computation distributed across brain regions drive behavior using linear models for encoding and decoding. However, we need more powerful models to extract more information from each brain region. And recent work has shown that increasing the scale of neural data improves performance on downstream tasks, such as encoding or decoding. However, current large-scale models focus exclusively on either encoding or decoding, limiting their ability to capture the bidirectional relationship between neural activity and behavior. So to bridge this gap, we introduce a multimodal, multi-task model that allows simultaneous neural encoding and decoding at scale. Neural encoding and decoding can be interpreted as modeling the conditional probability distribution between neural activity and behavior. In NES, we use a multi-task masking approach to model the conditional expectation of this distribution, as well as to encourage cross-modal and within-modality representation learning. This is achieved by alternating between neural behavioral within-modality and cross-modal masking during model training. We implemented NES using a multimodal transformer-based architecture. The first step is to use modality-specific tokenizers to convert spike counts and continuous behaviors into 20 milliseconds temporal tokens. We then convert discrete behaviors into sequences of repeated tokens to align with the temporal resolution of the continuous data. After that, we add temporal modality and session embeddings to the tokens, and tokens from all modalities are concatenated into a single unified sequence, which is input to the shared transformer encoder. And having a shared encoder makes sure that data from different modalities share a common representation space. We train NES by masking all tokens according to the masking schemes and then predicting them with modality-specific linear decoders. So similar to MTM, we again trained NES on a subset of the IBL repeated set dataset. But this time we included data from more animals for pre-training. And in our experiments to show the improvement from multimodal learning, we compare NES against a full-rank linear model, reduced-rank linear model, and a unimodal version of NES across 10 single sessions. And we evaluate the models using neural encoding and decoding tasks. For encoding, we predict neural activity using both the discrete and continuous behaviors. And for decoding, we benchmark the performance on decoding each single behavior using the for neural activity. So the bar plots clearly show that multimodal nets outperform the corresponding unimodal baseline. And also to show the benefit of multi-session training, we pre-trained nets on 73 animals and then fine-tuned it on each of the 10 held-out animals. And comparing multi-session nets with the single-session nets highlights the advantage of scaling neural encoding and decoding. And here are just some qualitative comparisons of the single-session and multi-session nets. So we also compare nets to two recent transformer-based models, POYO Plus and NDT2. So at the time of the comparison, NDT3 has not yet been released. And these models represent the current state-of-the-art in neural decoding. And importantly, each model has a different pre-training objective. POYO Plus performs multitask neural decoding of choice, block, will, and motion energy across all animals. And NDT2 performs self-supervised prediction of neural activity across all animals. And NES performs multimodal multitask masking across all animals. And we evaluated the fine-tuning performance of NES, POYO Plus, and NDT2 on the 10 held-out animals after pre-training on data from 73 animals. And we found that NES outperforms POYO Plus and NDT2 on ABL data across all the decoding tasks. And importantly, NES is also able to perform neural encoding unlike the other methods. So although NES is trained to perform neural encoding and decoding, we want to understand what additional information about the neurons is captured by the latent embeddings of our model. So we want to evaluate how well the embedding of a neuron predicts the brain region it is located. So to test this, we train a linear support vector classifier on the neuron embedding from the 10 held-out animals after fine-tuning. And we evaluate brain region classification using 5-fold cross-validation across all neurons. And we compare the neuron embedding from our pre-trained nets to single-session, unimodal, and multimodal versions of nets. And we found that the classifier achieves higher accuracy with neuron embedding from multimodal nets than from unimodal nets. And pre-training across 73 animals further improves performance with multi-session nets having the highest classification accuracy among all the methods. So surprisingly, the learned embedding from nets show emergent properties. Even without explicit training, they are highly predictive of the brain regions in each recording. So far, we've only scaled MTM and nets to cover animals and brain regions in the ABL repeated site dataset, which has a more limited scope compared to the brain-wide map dataset. Moving forward, we want to scale this foundation model to the full brain-wide map dataset. And additionally, we also plan to host a brain-wide map benchmark to fairly compare the performance of neural foundation models and improve our understanding of distributed neural computation across the brain. And finally, I want to say thank you for our amazing collaborators.